I'm C.M.Ciarleglio, and I'm an instructor at Johns Hopkins University, and I study circadian rhythms and developmental neuroscience. Today's presentation is going to be on circadian rhythms and giving you an idea of what I think is arguably the most important behavioral adaptation in the history of life on Earth. We're going to start by doing a basic background of circadian rhythms. Then we're going to compare human versus mouse model systems to show you how much we can learn from looking at other model systems like mice and flies. We're going to talk about the third of the basic properties, which is entrainment, then some anatomy, some molecular genetics, and then the consequences of circadian pathophysiology. At the end of this lecture, it's my hope that you'll appreciate the importance of circadian rhythms and have a basic understanding of circadian physiology. The reason that I think this is important is because a lot of professors that you're going to encounter in college probably aren't going to think this is very important, but it is. It touches almost every other physiological system, and in most cases, it's actually in command of those physiological processes. What is a circadian rhythm? According to the chronobiology textbook by Pat DeCourcy et al., a circadian rhythm refers to an observed biological activity that oscillates under constant environmental conditions with a period length close to, but not exactly equal to 24 hours. This is a very succinct definition. Some examples of this behaviorally might include eating, drinking, socializing, which in college is synonymous with sex, general activity such as running in animals like mice, the sleep-wake cycle, which is the most obvious phenotype that we see in humans and is the thing that is most talked about when people talk about circadian rhythms at all. Torpor, which is kind of like a hibernation state in birds and some other animals where their body temperature drops at night and they go into kind of a hibernation state. And then drug use. This is the drug abuse, the type of drug use that's bad, tends to behaviorally be under circadian control. On the molecular side, cell division is actually under the control of the circadian clock. And so when you have something like cancer going on, the circadian clock is not controlling the cell division as it should. It's in control of the actual checkpoints. Depression, which at the molecular level, and we'll talk about this a little later, especially when we do anatomy and the molecular genetics, depression is actually linked to the circadian clock. Hormonal regulation is all under the control of the circadian clock because the clock is located in the hypothalamus. Drug susceptibility, and this is of the good kind. If you had cancer and needed to undergo chemotherapy, any good oncologist worth their salt is going to try to titer your dose so that you have the fewest side effects possible. Those side effects actually have a lot to do with the division of those cells, where cancer cells are dividing rapidly out of control, and the circadian clock is usually under control of all the rest of the cells. If you find the time of day in which your normal cells are not actually dividing, you have fewer side effects by giving the chemotherapy at that time based on the circadian clock of the person. There are three main properties of circadian clocks. The first is the most obvious. It's about 24 hours because the length of the day is about 24 hours long. The second one is less obvious, and we're not going to talk about it at all, and that means that it's temperature compensated. So anyone who studied chemistry knows that as you increase the temperature of a reaction, that reaction tends to go faster. Circadian clocks don't do this. They continue to run at an around 24-hour period, regardless of what the temperature is. We don't know why this happens. The last one, which is the thing we're going to most focus on, is entrainment. The fact that some sort of stimulus from the outside world will actually entrain the circadian clock and set it every day so that it's not, as we would call, free running, and you'll see that in a moment. So what is entrainment? What is the relationship between the outside environment and the inside? In this classic drawing from the Moreed book, The Clocks That Time Us, published in 1982, it actually draws out all of the relationships you would see. This here is the Zeitgeber, the German word for time giver, and that is usually light. So the sun comes up, so that's dawn, and then stays up, and then goes back down, and that is dusk. And on a typical equinox light cycle of 12 hours of light, 12 hours of darkness, you would have this normal oscillation. And this is very regular, and it's been doing this since the dawn of Earth. In response to that, this is the rhythm. And so what you see is that the rhythm of whatever it is you're measuring will increase at one point during the day and decrease at a different point during the day. The measurement between 2 points is called the period, and that's denoted here by the Greek letter tau. So from peak to peak is the period. You also have the amplitude. The master oscillator, which would be the sun, has a capital phi, which is the phase at which, say, dawn occurs. And the slave oscillator would be the small Greek letter phi, the phase at which the peak of this rhythm, whatever you're measuring, occurs. The relationship between them is psi, which is phase angle. And that's actually the point of a circadian rhythm. The whole point of having this particular adaptation is that living things need to maintain their relationship, the timing they have with the outside environment. The early bird gets the worm. You do not want, if you're a bird, you don't want to wake up too early because you won't find anything to eat. And you don't want to wake up too late because you'll have missed it. So you need to be right on time. The Zeitgeber, as I said, is a time giver, and it does not have to just be light. What are the circadian rhythms that you see, especially in humans? So here are some examples. On the left, you see different measurements, what we call actograms, for different phenotypes in humans. Your sleep stage, for example, back when it used to be measured as 5 different stages, now it's only 4. And you see that sleep tends to only occur during the nighttime, which is denoted by this black block. Sleep only happens during the nighttime, but during the daytime, not so much. Temperature. Your temperature actually increases to about 37 degrees Celsius, that's about 98.6 degrees Fahrenheit, during the daytime, and then drops precipitously at night. Your growth hormone is only released right after you fall asleep. So if you happen to be in your teenage years, in puberty, then it's very, very important for you to actually get sleep and get to sleep early and stay asleep for a long time, because that's the only time the growth hormone will actually be released. If you go to bed really late because you have projects or you're playing video games, then you're actually pushing off the release of that growth hormone and giving yourself less time for it to have its impact. If you want to grow, you need to sleep. Cortisol is the stress hormone, and that also tends to wake you up in the morning. So you actually see that it peaks in the morning and then falls off as the day goes on. Lastly, potassium excretion only happens when you're awake, because I don't know if you're like me, but I don't like wetting the bed, and the only way to get rid of potassium is by urinating. So no urinating at night. That's mostly during the day. Over here, these are actual measurements that deviate from the normal average over the course of the day. Things like rectal temperature or catecholamines. Catecholamines are things like dopamine or norepinephrine, which will be covered in this course. Your reaction time increases. Meaning you slow down in the very early morning. And this makes a lot of sense, because if somebody came in at 3 in the morning and said, hey, let's go do some math, I don't think that you'd be very good at it. The birth rate also happens at about 2 a.m. It tends to peak. And so does the death rate. So you really don't want to go run a marathon at 2 in the morning, just so you know for the future. So I want you to think of an undergraduate's life on the clock. So you are in the situation where you're taking a course at Johns Hopkins University. You're an undergraduate, and this would be a typical light cycle. So you would wake up pretty late. Adolescents tend to wake up much later than they will much later in life, and much later than they do when they're children. But when you wake up, by that point, you can't really have breakfast. It's too late for breakfast, and it's a little too early for lunch, so we'll call it brunch. Then you have classes, and you have this thing in the middle called a postprandial dip. That postprandial dip, a lot of people think, is due to food from your lunch. It actually isn't. It's genetically programmed for you to take a nap, so a siesta is actually a good idea. You have those classes, you might go back to your dorm, have dinner, and then you do some socializing. And that socializing does a lot to actually change the architecture of your circadian rhythm, so that you're going to bed later, and therefore your sleep cycle is much later. Let's compare this to something that we could actually study and gain information from, like a mouse. If you think of a mouse as a clock, there's two different types. There's breeders and there's experimental mice. Okay, breeders will wake up at dusk, very different from us. We're diurnal and they are nocturnal. And as nocturnal animals, they wake up at dusk and continue to run or be active throughout the night. They are nocturnal. All right, so they might eat, they give birth at around the same time we do, and then they fall asleep. Now, one of the problems with this is, and one of the reasons I think that you need to convince your future professors that circadian rhythms are really important, is because this really screws up experiments a lot. People don't tend to realize that mice are nocturnal and their normal activity period is not during the day. So when the annoying student comes in to test a mouse's memory at noon, it's the equivalent of me coming in and waking you up in the middle of the night to test your math skills. It doesn't make a lot of sense. The other type is called an experimental mouse. Again, it wakes up at dusk, realizes it has a wheel, and runs on it all night. Mice, for some reason, a lot of the small rodents, they love wheels. And it's a great way to actually measure their activity, which you can do in an actogram or an actograph, as you see here. So here are two different versions. This is the same mouse running on a wheel and also being measured with an infrared sensor. And what you see is the black tick marks are the activity. So this would be a rotation on the wheel, and these little ticks would be beam breaking in the infrared. And so you can compare them to each other. The yellow part is when the lights are on, and on the y-axis here are the number of days. So day 1, day 2, day 3, day 4, day 5, etc., etc. What you see is that their activity actually begins right at dusk. Their phase angle is essentially 0 with dusk. They don't tend to happen before or after. But if you shift that 6 hours, as if you're going from the eastern seaboard of the United States to London, England, a 6-hour advance, as you see here, I've advanced the light cycle a certain way, then it actually takes them a while to catch up. That's called jet lag. And you could do this in a lab without actually moving any animals at all anywhere. You just need to change the light cycle and they will actually experience jet lag. If you were to then put them into constant darkness with no light cycle, they will free run with a specific period that is a property of their own internal clock. And there's always a little bit of variation from mouse to mouse. So it's important to note, though, that you do actually get entrainment again after the jet lag is over. So that when you phase delay them again, they experience jet lag again until they eventually come up with a proper phase angle of entrainment, showing that they're showing a correct true circadian rhythm. These are really useful to look at. So how does this entrainment happen? How does jet lag happen? In general, the most important Zeitgeber is light. So sun hits the retina of the eye, goes into the brain, is processed into some sort of behavior. So in the retina, you have a bunch of cells. You have actually 3 different photoreceptive cells, rods, cones you may be familiar with, but they're actually also intrinsically photosensitive retinal ganglion cells, which produce a pigment called melanopsin. That melanopsin was first discovered by Ignacio Provencio and then located in the retina of mammals by David Burson and Samar Hattar. And Samar Hattar at the time was at Johns Hopkins University. This is in a mouse, so mice tend to have a shorter free-running rhythm than humans do. Humans tend to be the other way. If you give a pulse of light during the daytime, it doesn't actually shift them at all. If you could see this, see how they're running every day up until the day of the pulse, and then after the pulse, they continue to run on their normal free-running rhythm because the pulse hit them during what we call their subjective day. And that makes sense because you usually do see light during the day and you don't need to adjust to it. But if that light occurs right as nighttime's supposed to begin, you will get this, what we call a phase delay. And if it's significantly into the activity period, it will delay a lot the next couple days, and then it'll resume its period. And you can actually measure that as a phase shift. And then you can plot these on a graph like this, which is called a phase response curve. The phase response curve has this characteristic dead zone during the circadian day, because light shouldn't affect your clock. But if you get some light early in the nighttime, it tricks your clock into thinking, oh no, I guess I'm running too fast because it's actually much earlier than I thought it was. Contrary to that, if you give a pulse of light in the late night, so almost morning, it will do the opposite. It will actually advance it and convince the clock, I'm anthropomorphizing here, but it'll convince the clock that it's actually running too slow and needs to pick up the pace. And so you will see an advance here. Where is this all occurring? The suprachiasmatic nuclei of the hypothalamus are located in the anterior hypothalamus just above where the eyeballs send their projections to the brain, where they cross at something called the chiasm. So in a mouse, it would be located right here. So this is a ventral view of a mouse brain. It is located right here. This is the optic chiasm with the optic nerves coming in and the optic tracts going back out. If you were to do what we call a coronal slice, a slice this way, and look at the inside of their brain, you would see these two bulbs facing you, which can be subdivided into what we call the shell and the core. And this is the site of the circadian rhythm in mammals. This circadian clock is the master circadian clock that controls all the other clocks in the body. And it is tightly linked to other important nuclei in the brain, such as the raphe, which produce serotonin. So you have a projection from the eyeball, which is glutamatergic. It produces glutamate, the main excitatory neurotransmitter of the brain. And that will actually phase shift the circadian clock. And this will interact with other nuclei in the brain. The important one that I think that is interesting is the raphe nuclei, because the raphe produce serotonin, and serotonin is linked to your mental health and mood. So if you ever feel sad during the winter, it might have something to do with your circadian clock actually interacting with your raphe nuclei. Within the suprachiasmatic nuclei, or SCN as we call it, there are 10,000 very small, very closely packed cells in each of the nuclei. They are master oscillator cells. And the way that works is by the expression of certain circadian genes that are really important. So it's a very simplified cell with a massive nucleus and very little cytoplasm. Neurons don't look like this at all. RORA activates the expression of BMAL. Clock is also constitutively expressed. Those transcripts go into the cytoplasm and are translated by ribosomes. The resultant proteins will dimerize and then be phosphorylated so that they can go back into the nuclei and activate the expression of PER, cryptochrome, and reverb alpha. In our animals, that turns the cell green. Ribosomes will translate these into proteins. Period 1 will go and bind to cryptochrome. Casein kinase will go and phosphorylate period, which will allow it back into the nucleus, stopping its own activation. Reverb alpha stops the activation of BMAL1, and that takes about 24 hours. And so these genes are expressed at certain times of day, regardless of the Zeitgeber status. So what you see here are the expression of genes over the course of the day. So here is 0. So ZT0 is the onset of light in mammals, in diurnal mammals. And ZT12 is the offset, so dusk. And what you see is that the period genes, there's 3 of them in humans, will increase during the daytime, whereas BMAL only increases at night. And this continues even in the absence of any Zeitgeber. So you can see a drawing of it of relative expression up here. As PER1, PER2, and PER3 are being expressed, you also get an increase in the activity of the neurons electrically in that part of the brain. So here's a simplified version that we've drawn for a textbook, which was recently published, where, again, BMAL1 is activated by RORA. BMAL1 goes out, is translated into a protein. That protein goes and activates the expression of PER and cryptochrome and reverb alpha. Those things trimerize and come back in and turn off their own expression. This is a classic transcription-translation feedback loop. In 2017, Every circadian biologist was excited to learn that their field had finally been recognized by the Nobel Committee when Jeffrey Hall, Michael Rosbash, and Michael Young were awarded the 2017 Nobel Prize for Physiology or Medicine for their discoveries relating to the genes that control the circadian rhythm. We were very excited about that. These genes were first isolated in flies. While screwing up circadian rhythms can have broad implications for all of your physiological systems, since they're almost all under the control of the circadian clock, some specific ones are actually identified as circadian disorders. There are several defined here. If this is your normal sleep pattern where the black blocks are actually sleeping on this particular actogram, if you have delayed sleep phase syndrome, DSPS, you have trouble falling asleep until very late at night, and then a lot of trouble waking up in the morning. That could be delayed sleep phase syndrome, and it actually has a genetic component. The opposite of that is something called advanced sleep phase syndrome, where you or your family can't help but fall asleep very, very early at night, and then wake up in the middle of the night because you can't sleep anymore. Familial advanced sleep phase syndrome is actually caused by mutations in PER2, a period gene, and delayed sleep phase syndrome and non-24 are actually caused by PER3 and casein kinase 1 epsilon mutations. There is also this other thing called non-24, which means that you free run like a mouse in darkness. You free run through life. This happens a lot in people who are eye blind. Meaning that they don't have functional eyes anymore, and their brain is continuing to process circadian rhythms where, since they can't see the Zeitgeber light, they're forced to free run through life. This could be quite debilitating. So what have we learned in this circadian lecture? First, the basic properties. You've learned that circadian rhythms are about 24 hours, that they're temperature compensated, though we didn't talk about that much. And you learned that they are entrainable by Zeitgebers. We've looked at the difference between diurnal humans and nocturnal mice and seen that mice are actually still a very good example of how circadian rhythms work behaviorally and molecularly. We've looked at entrainment and seen that light is the primary Zeitgeber. We looked at the anatomy and found that suprachiasmatic nuclei are the seat of the circadian clock in mammals, and they're located in the hypothalamus above the optic chiasm. The molecular genetics were interesting because they also won the Nobel Prize for Jeffrey Hall, Michael Rosbash, and Michael Young for the discovery of these genes, especially the period gene. And we now know that there are consequences of circadian pathophysiology. You should also now, of course, appreciate the importance of circadian rhythms, hopefully, and have that basic understanding and maybe be able to convince your future professors that circadian rhythms are worth considering when you're designing an experiment. I'm C.M.Ciarleglio. Thank you very much.